Another 1 minute. So let me kindly introduce the facilitator of the day for this morning session, which is none other than our own ecologist from Forest Research Center Sepilok. Ladies and gentlemen, please welcome the handsome Dr. Ruben Nilos. Dr. Ruben, the floor is yours. Over to you.
Oh, thank you. Thank you so much, Beatrice. You make me feel like I want to howl. Oh, anyway, selamat pagi dan salam sejahtera. A very warm greeting to you all, and thank you for allowing yourself to be with us today. Through the Heart of Borneo Scientific Expedition Project component, Sabah Forestry Department has organized a scientific study at Mensalong Forest Reserve from the 29th July to 4th of August 2019, with participation from University Malaysia Sabah and Sabah Park. My name is Ruben Ilus. I will facilitate the morning webinar session, and my good friend, aka Nemesis, Dr. Arthur Chung, will facilitate the evening session. Welcome to Sabah Forestry Department 4th webinar of the year with the theme Mensalong Forest Reserve, Heart of Borneo Scientific Expedition Webinar 2020. There will be 13 presentations in this webinar, and I hope you all could be with us throughout the sessions. There's a saying, we have the choice to use the gift of our life to make the world a better place. And I believe we are some of the few here. If not, why are we participating in this webinar? To initiate our webinar today, with great pleasure, I would like to invite Mr. Frederick Cogan, Deputy Chief Conservator of Forests, Planning and Management, Project Leader of Heart of Borneo, Sabah, to deliver his opening remarks. Tuan Fred, the line is yours.
Okay. Thank you, Mr. Facilitator, for the kind introduction. And a very good morning to all, especially to our colleagues from the Forestry Department as well as from Parks UMS, who has been a very strong supporter of this expedition work. Not to mention also the relevant government agencies that have supported over the years. Well, my task today here this morning is to say a bit about HoB. I think everybody knows HoB ever since its inception in 2007. It has been quite some time that we have implemented the initiative itself, and I think my guess is that everybody here in this room are very well aware about the objective. The objective for Heart of Borneo, to sum it up, it's all about sustainable development. And of course, in our course, in order to implement Heart of Borneo, there are 5 pillars. I think just to remind you that there are 5 pillars in Heart of Borneo initiative itself, which looks at transboundary management, protected areas, sustainable natural resource, ecotourism, and capacity building. And for Sabah itself, we have 3 main priority areas. Basically, looking at maintenance of forest connectivity, which is very crucial. It's all about linking up the fragmented forest reserve or protected areas in the state and identify where are the crucial link that need to be protected in the whole Sabah landscape. And of course, by doing so, we will be looking at what are the opportunity to enhance transboundary cooperation, not only within Sabah, but we also look at what are the transboundary cooperation with Sarawak, interstate, as well as international with Brunei and Indonesia. And the implementation of HoB, as we all know, over the last more than a decade, focus a lot on the totally protected areas. I think we have set a target in Sabah to achieve TPAs about 30% by 2025. To date, we have achieved about 26%, remaining about 1.9 million hectares, and we still have about 300,000 hectare remaining area to be identified. And in doing so, there are quite a number of programs because HoB is not only about keeping up TPAs, but there's a lot of other activities associated with it. For example, protection, expedition like this. And over the years, I think we have, thanks to the federal government, to KEXA now, that they have invested about nine Malaysia plan up to now, total about 60, 11 is about 54 million. And out of this, coming up to 2 million has been invested into expedition like this. And I was informed that we have conducted expedition in about 37 forest reserves in the state. So I think that is a remarkable achievement expedition. Why are we doing this? Because there's a lot that still need to be learned, to be explored. And what is more important about this expedition, from the perspective of the management, is that we need to identify what are the key conservation targets. One, and then after that, we identify what needs to be protected and what sort of action or activities or that can be introduced in order to enhance its protection. That is looking at the protection of the areas or protected areas of forest reserve. And beside that, I think we do this expedition to 37 forest reserves that has been covered. I'm sure that has already enriched our knowledge about what we have in the state. And over the years, I think the new species or even rare species have been identified, and I think that is a testimony that on the achievement. And I think I think it will be interesting to listen to the 13 presentations today on what has been identified, or probably there may be some new species. But then again, these are all very important, yeah. But then again, we need to translate this and how this will benefit to the society at large. Because in the conservation world, I think we all understand why is it so important that we protect and conserve. But then again, I think we need to translate it to the community, to the public, socialize a lot more, and in fact, what is the focus of HoB next? I would call it HoB next, or maybe expedition next. Yeah? Is that the effort will focus not only on protected areas of forest reserve, but look into other landscape outside of protected areas of forest reserve. That means looking at areas in the state land, title land, community land, and the concept, in a way, it is very much in terms of mainstreaming biodiversity conservation. What we have learned, what we have acquired through all the 37 or even Mensalong, the lessons learned, and how can this be brought into the other landscape? How can we assist the community? How can we assist the other government agencies in order to come up with a more sustainable practices, for example, land use planning, for example. So this brings about the focus of HoB next, or expedition next, the focus of areas which is areas outside. And I'm glad to inform that there are quite a number of projects online. Hopefully it will be approved soon. We will implement it next year as a start of the next 12 Malaysia plan, focusing on areas such as dealing with communities and so forth. So there's a lot of socialization that need to be done. I'm not too sure how effective will webinar outreach, but then I think this is something that may be under the initiative here to look at all other avenues or options on how to reach out. In fact, very important that we have to increase our socialization about this, expand awareness raising and so forth, not only within our sphere of intellectual here, researchers, etc., but then reach out to the community themselves. And I think this is very crucial. Unless they understand or appreciate what we are doing, appreciate what the role of conservation, I think we're bound to have maybe more obstacle in terms of our effort in order to promote conservation. But then again, we have always, as far as the department is concerned, forestry department, we have always alluded to all saying that conservation is not only about forestry, it's not only about the forest, but in fact it is supporting a bigger economies. Just imagine, without forest, there's no ecotourism. Without forest, there's no water. Without forest, there's no other big initiative, big projects, etc. And I think this is very important. That is why, under the HoB initiative, I think the key is that it is about bringing sustainable development. And I think it is about time that we move to the next agenda or era, which is HoB next or expedition next. Focus on the other frontier, not only. Because there's a lot more. We have covered 37 forest reserve or protected areas. And I think there's a lot more, even within, that need to be covered. But this HoB next will focus on the other landscape as well. And I think this is also very crucial, very much in time with the initiative, with the ongoing initiative by the Forestry Department to look at the state as a whole in terms of a master plan for forest master plan, whereby the concept of conservation approach will be to rationalize land use, protect whichever the best forest left and there, and then look at the other opportunities in terms of non-net loss, net gain, PES, etc., etc. And expanding the concept of TPA not only on totally protected areas, but also to look at other concepts such as the community conserved areas. These are the things that should look at. And what can expedition contribute to that? I think there's a lot. Yeah? And there's a lot of engagement with community, especially if it is involving their land, native land, or it may be state land. Yeah, they are quite under, I'm sure, quite a number of interesting areas, not only within the protected area, forested areas that are important to be conserved. But then again, as we say, when we deal with community, sustainable livelihood comes in. So maybe the elements about when we look at expedition next in terms of this landscape, maybe we will have to look at what are the non-timber forests produced that can be commercialized or can be utilized, maybe by the community or even commercialized at the commercial level. I mean, these are the new frontier. I mean, we have not, correct me if I'm wrong, researchers, that we have not developed non-timber forests produced as far. Maybe because there's a lot more need to be learned. And also, there is also the fact of market and etc. etc. So I think with that, that is a brief snapshot about what is HoB next, or what is conservation next, what is expedition next. So I hope that you can expand your horizon to that new landscape, new, in order to reach out to a wider public or even to the local grassroots people, grassroots on the ground. So I think with that very much, I would like to take this opportunity, thank all the participants here, and also especially those who have involved in the expedition of Mensalong. We will hear a lot more about Mensalong today, and of course, I'd like to congratulate the organizing committee for having organized this webinar. We're supposed to have HoB every year, but I think we have changed that mode once every 2 years in order to have more output. But then I think due to the current situation of COVID and so forth, I think we cannot avoid having this webinar. One of the effective mechanism or mode in order to reach out But then again, I think we will have to think hard on how to reach out to the people. And I think thank you to all the speakers, and I hope all will have a very fruitful session, and we look forward to receive any input or recommendation that you may have in order to continue implementing the HoB in the state of Sabah. So I think with that, Mr. Collaborator and MC Elni, thank you very much for inviting me to grace this morning webinar. On behalf of the Chief Mashur, I think we wish you all a fruitful session. Thank you.
Thank you, Tuan Fred, for the overview remarks on HoB vision, strategic plan, and activities. Copy that. Now we're moving on to our special keynote presentation. Cherish the natural world because you are part of it and you depend on it. With honor, I would like to invite Mr. John Sugao, the Chief Commander of HoB Scientific Expedition, Curator of Sandakan Herbarium, Program Leader of Forest Biodiversity and Conservation Research in FRC. Hello, partner. The line is yours.
Okay, let me share my screen, my PowerPoint. Okay, once again, thank you, Dr. Ruben. Good morning to all. In this presentation, I'll give an overview of the Mensalong Forest Reserve Heart of Borneo Scientific Expedition. And this overview includes some introduction or information about the reserve. And secondly, we'll highlight on the activities conducted during the expedition. Firstly, let's talk about location of Mensalong Forest Reserve. The Mensalong Forest Reserve, which cover an area of about 8,000 hectares, is located in the 3 districts, namely Kutamarudu, Beluran, and Ranau district, but administered by the Kutamarudu District Forest Office. And the reserve is actually formerly part of Linkabau Forest Reserve, a Class 2 Commercial Forest Reserve, then was re-gazetted as Class 1 Protection Forest Reserve in 2016. And this forest reserve share approximately 5 kilometers of its boundary with Kinabalu Park in the west. So this is very important as a corridor connecting Kinabalu Parks and the larger part of Linkabau Forest Reserve. And the reserve is easily accessible by sea route, which is Ranau Kutamarudu Highway. This map shows the topography of the reserve, and most of the forest reserve areas are on the highlands, except for the eastern and northern western side. And the altitude ranges from 250 meter to 1,100 meter above sea level. And the reserve serves as a water catchment for 2 main rivers, namely the Sugut River, which flowing to the Sulusi and Sungai Bongan to the Marudu Bay. The soil in this reserve is mostly classified as Crocker Association with dominant soil unit Otic Acrosol district, Chromic, Cambisol, and Lutisol. And I believe our soil expert will explain more on this. The original vegetation of the reserve consists of mainly upland mixed dipterocarp forest with some lowland mixed dipterocarp forest and small patches of montane forest in the middle. But currently, the forests are considered disturbed due to logging activities in the past. This is the photo of the base camp, which was set up at Kampung Teruntungan, a kilometer south of the reserve. Apart from Kampung Teruntungan, there are 2 more villages. in the vicinity of the Mensalong Forest Reserve, namely Kampung Monggis and Kampung Surun-Surun in the north. On the screen is the group photo of the participant, and there were 81 participants involved with participation from Sabah Forest Department, University Malaysia Sabah, and Sabah Parks. On the first night at the base camp, expedition leader, myself, welcomed the participant to the scientific expedition, and at least 15 groups of researchers from various disciplines conducted the survey during the 5-day expedition. And representative of the surrounding villages was also present and given opportunity to speak about their view on the expedition. And in the photo, we can see the village head of Kampung Turuntungan giving his view and giving his welcoming remarks. Now, I'm going to highlight some of the activities conducted during the expedition. And this photo taken at the side of Ranau Kutamordo Highway, which cut through the Mensalong Forest Reserve. For the forest assessment, Mr. Muhammad Aminul Faiz and team of SFD studied the forest structure and composition of the forest reserve. And 2 forest ecosystems were sampled throughout the study, namely the lowland mixed dipterocarp forest and upland mixed dipterocarp forest. And a total of 8 permanent sampling plots were established. The findings were forest structure and tree species composition among these forests are varied, and although the vegetation are impacted by previous anthropogenic and disturbed by activities in the past, the ecosystem are well regenerating, and forest ecosystem in the area are of high conservation value as they provide viable habitats for many fauna and flora species. Plant diversity survey and botanical collection were carried by me and my team from Sandakan Herbarium, and also Richard Majapun worked on dipterocarps and recorded some 34 dipterocarp species. And this study recorded 821 taxa from the reserve and found the forest in Mensalong Forest Reserve still contain high conservation value plant species. Dr. Kartini Saibi from Faculty of Sustainable Agricultural, University Malaysia Sabah conducted research on Araceae, which is the family of taro. A total of 8 genera comprising of top species are recorded, and of these, at least 2 representing new species. Easter Day of Sabah Forest Department did the soil assessment during the expedition. The study showed that soil in the reserve is classified as, of course, a crocker association, and these soil units are generally shallow or to moderately deep and highly erodible, well drained, with soil color varies from dark brown to yellow. The texture ranges from loamy sandy and sandy clay loam. Mrs. Loiza Muajakim of University Malaysia Sabah studied dissolved organic carbon in the soil. And the findings of the study indicated that the total organic carbon TOC varied between forest types and across soil depth. And this suggests that the impact of soil organic carbon loss due to land use change could be more extreme to, for mountain forest. Survey on fungi were carried out also by Ms. Vianney Paul of the Sabah Forest Department with Dr. Jaya Silan of UMS and his student. Among the fungi that can be found around the lowland and highland of the forest reserve area are gill fungi, bolitis, pivot, bracket fungi, as well as the star fungi. I believe Vianney will explain later on this. Again, Mr. Aminur Faiz of Sabah Forest Department, together with the team from the Sabah Parks, led by Johannes Gempoya, were also researching on wildlife, and a total of 22 species were recorded during the field survey. detected through camera trapping methods and recorded along the transect. Among the wildlife recorded are leopard cats, bearded pig, manjak, long-tailed porcupine, etc., etc. Hubert Peter of the Sabah Forest Department and team surveyed the avifauna of birds, and a total of 101 species from 37 families were recorded, and there were 10 species of Bornean endemic detected, and the blue banded pita and the Kinabalu serpent eagle are categorized as vulnerable in the USN Red List. Mr. Pangeran Salan of SFD and team conducted survey on frogs and toads. A total of 401 individuals were caught, consisting of 16 species belonging to 6 families were recorded, and top Bornean endemic species were recorded during the survey. And the endemic Anoran species recorded during the survey provide very significant information to enhance the conservation of the Mensalong Forest Reserve. Mrs. Nur Safikah Kamal of Sabah Forest Department also did some work on the fishes of rivers within the reserve. A total of 559 fishes grouped in 25 species were captured, and the most abundant species was Tortambra in the area with clear water and moderate swift flow. Mr. Harold Hafiz of Institute of Tropical Biology and Conservation, UMS, also sampled parasite of fishes. Dr. Arthur Chung, Raji Zapir, and entomology team of SFD surveyed the insect fauna, which include butterflies, moths, beetles, and dragonflies. And the study shows a moderate high in terms of diversity when compared to other forest reserves in Sabah. And from the survey, there were 9 Bornean endemic species recorded. The late Dr. Bakhtiar Yahya, with kept in the photo, and his assistant, Mr. Zakaria Barabag, both from Institute of Tropical Biology and Conservation, UMS, work on ants. For our information, the late Dr. Bakhtiar was an associate professor with the UMS and led the UMS team during the expedition. But unfortunately, he passed away last year, 24th February 2019, due to dengue fever. So, semoga rohnya dicucuri rahmat oleh Yang Maha Kuasa. Beside conservation, social science was also conducted by Mr. Jeffrey Lapungan and team. And based on the findings of the study, almost 60% of the local community are still dependent on the forest resources. And this study also explored other aspects such as awareness on forest importance and perception of communities on the past government and non-government activities. Nature tourism is another aspect that was given emphasis in this expedition. Mr. Jerilan Jaga, also from Sabah Forest Department, surveyed the potential size and product of nature tourism. And according to him, developing the Mensalong Forest Reserve as an ecotourism site is an opportunity that is beneficial to the local community and the surrounding areas. Though threats from human activities cannot be eliminated, but he said proactive measures can reduce the impact. So, in conclusion, the researchers will explore the forest in Mensalong Forest Reserve. Hence, such data would be very useful as a basis for the formulation of conservation area management plan for the reserve and surrounding areas. And this also signifies a very good participation from researchers from other agencies, as always encouraged, and to fulfill one of the objectives of the Heart of Borneo, that is Heart of Borneo Scientific Expedition. So with that, thank you, ladies and gentlemen.
Ben, can you unmute your microphone?
Okay, sorry.
Yeah.
Sorry. Thank you, John, for the
Welcome.
description of the site, participant involved, and the studies that have been carried out. I wish I could be there. Really make me feel jealous. Anyway, once again, for the presenters guideline, before we start with the morning session, I would like to remind the presenters again that you are allocated 10 minutes for your talk. Use the time wisely. The secretary will ring the first bell to indicate that you have 3 minutes left. And the second bell indicate that you only have 1 minute left. I guess that's where you should conclude your talk. So if any of the participants of this webinar have any question for the presenter, again, please use the Q&A tab button located under the Zoom presenter presentation window. Please don't use the chat window to forward your question. For the morning sessions, we have 7 presentations, and we are going through all the talk without taking a break. And after the 7 presentation, we're going to have 30 minutes question and answer session. Okay, I hope that's all clear. Shall we start, Secretariat? Dr. Arthur's Angels ready? Give me the thumbs up. I want to see the thumbs up. All right, okay, let's start. Like the great John Mayer used to say, the clearest way into universe is through the forest wilderness. With great pleasure, I would like to invite our first presenter, Mr. Muhammad Aminul Faiz bin Swiss, the Secretary of HoB Scientific Expedition, forest ecologist, one of our best drone pilot to deliver the forest assessment in Mensalong Forest Reserve. Hello, Captain. The line is yours.
Okay. Thank you, Dr. Raven. Let me share my slide first before I start my presentation. I hope you all can hear my voice clearly. Okay, good morning, everyone. You can call me Faiz. I'm about to present a research that was conducted during the scientific expedition. The title of our presentation is The Assessment of Forest Ecosystem in Mensalong Forest Reserve, Kota Marudut, Sabah. This research cannot be materialized without these awesome members. There are Dr. Ribenilus, General John Sugao, Sandy, Petrus, Darren, Paiji, Jameson, Raisno, and Joel Tawat. This presentation comprises of 4 sections. The first section is objective, followed by methodology, result, as well as discussion, and the last section is conclusion. Our objective were to describe forest ecosystem in Mensalong Forest Reserve. We also want to establish permanent sample plot or PSP for the statewide biodiversity monitoring efforts. Moving forward, the methodology involved the establishment of 8 permanent sample plot in the reserve. We also performed 2 statistical analyses, namely importance value index and cluster analysis. Next, as I have mentioned before, the slide summarizes how the PSP is established. It is borderless and circular in shape. It also involves tree identification, data recording, tree enumeration, and labeling. Okay. We have set up a total of 8 permanent sample plots in the reserve. Most of them focus on the eastern part of the Mensalong Forest Reserve. So for result and discussion, Mensalong Forest Reserve comprises of 3 natural vegetation, namely lowland mixed ditrocha forest, upland mixed ditrocha forest, and lower mountain forest. Due to inaccessibility issue, we couldn't reach the western part that has lower mountain forests. So there are no permanent sample plot established in that area. Figure 2 is the output of cluster analysis. We can see that there are two major clusters. The first cluster is upland mixed dictrocarp forest, and the second cluster belongs to the lowland mixed dictrocarp forest. Okay, the lowland mixed dictrocarp forest is located below 509 meters above sea level. We found a number of disturbance signs in this forest, especially large canopy gaps, tree stumps, and abundance of pioneer species. This forest is dominated by dictrocarp with relative dominance of 28.67%, followed by Euphorbiaceae and Moraceae. At species level, the most dominant species was Shorea johariensis and Parashorea malanonan. One of the pioneer species that are common in this area was Macaranga pearsoni of Euphorbiaceae. The upland mixed dictrocarp forest occurred at higher elevation. It is also disturbed due to the anthropogenic activities in the past. As reflected by its name, this forest is dominated by Dictrocarpaceae, Myrtaceae, and Fagaceae. At the species level, Shorea obscura, Cesium sp, Lithocarpus are among the top dominant species. We observe a number of threats to the forest ecosystem in Mensalong Forest Reserve. Secondary vegetation are common in this area, as I mentioned, due to past anthropogenic activities. Fire spread faster in this forest due to the simple forest structure. Let's take a look at the image. You can see a spot, you can spot a column of smoke of urban burning. We are not sure whether this happened outside or inside the forest reserve, but the threat is real. Through this study, we suggested few measures or mitigative measures for the mentioned threat. First, the enhancement of fire prevention program. This could be done by additional fire towers, updating the current forest fire trend. Second is monitoring forest dynamic. We could do this by prioritize additional permanent sample plot so that we can do a robust analysis to monitor the forest dynamics over years. And since Mensalong Forest Reserve is in the vicinity of local villages, this local community play a critical role in conserving the biodiversity of the reserve. So environmental awareness, continuous environmental awareness should not be. As conclusion, there are three types of forest ecosystem in Mensalong Forest Reserve, but since they are impacted by the previous anthropogenic disturbance, there are no more disturbance at the moment. The ecosystem are well generating. In fact, the forest ecosystem has a high conservation value because they provide viable habitats for a wide range of faunal and floral species. I think that's all. Thank you very much.
Thank you, Faiz. Hello. Thank you, Captain Faiz, for sharing the living peoples of Mensalong Forest Reserve. Wonderful arrow shots, I would say. If you have any question for Faiz, please don't be shy. Use the Q&A tab located below the Zoom presentation window. Please don't use the chat window. Okay, moving on to the next presentation. There's a saying, all life on earth emanates from the green of the plants. With honor, I would like to invite again the second presenter, Mr. John Sugao, to deliver plants of Mensalong Forest Reserve. Hello, partner. The line is yours.
Hello. So let me share my screen first. Okay, thank you once again, Dr. Ruben. Once again, I appear to present or to share the finding of our study on diversity and conservation status of vascular plant in Mensalong Forest Reserve that we conducted last year. And this paper is co-authored with my colleagues as from FRC, as can be seen on the screen. As for very short introduction, in terms of flora, previously there is no record of botanical collection from Mensalong Forest Reserve. Hence, the objective of this study are first to determine the plant diversity, want to know the plant diversity there, and to identify conservation status of plant species and to identify high conservation value plant species, and eventually to provide flora data and recommendation to the management for the formulation of FMP or conservation area management plan of the reserve. For the methods, we enumerated all the trees of 10 cm diameter and above at breast height within the 8 numbers of 20 meter radius circular plots. And for information, we use or share the same plots or permanent sample plots, PSP, with those used by the forest assessment team. We also enumerated and collected all vascular plants within the plots, made general collection using SAN number, and the Dipterocarp team did survey on Dipterocarp species. And for the plant identification, we recorded all species that we could identify in situ or in the field, while those unidentified plants, vouchers were taken and brought back to the Sandakan Herbarium for further identification. And lastly, we compiled all the data to produce a checklist of plant taxa recorded from the survey. And this screen shows how we identify trees and collect plant sample in the field. And picture on the left hand side showing our tree finder identifying a plant or trees using binocular. And normally we identify tree species by looking at the leaf characters and of course the bole character and others. And for the difficult species, we collected plant samples using extendable poles of fallen leaves of the trees on the forest floor. We pressed the samples and brought back to the herbarium for further identification by matching them with the existing herbarium specimen. On this screen, to determine the conservation status and legal protection of plant, we refer or consult various references. For conservation status, we refer the IUCN release. And for the legal protection of plants, we refer Sabah Forest Enactment 1969 and Forest Rules 1969 and 68, Forest Rules 1969, Sabah Wildlife Conservation Enactment 1997, and appendices in the Convention on International Trade in Endangered Species of Wild Fauna and Flora, CITES. And for tax endemism, we refer various related taxonomic account. So now let us look at the result of the study. A total of 821 taxa from 117 families were recorded from the study site. And the most diverse is plant from the Angiosperm dactylon group, represented by 89 families with 713 species. Monocotyledon 13 families with 73 species, followed by fern group represented by 13 families with 27 species. Leucophytes or fern allies, 1 family with 5 species. And lastly, by the gymnosperm represented by 1 family with 3 species only. This graph shows the 10 most species families in decreasing order, preceded by the Rubiaceae with 50 families, and the least are the Maliaceae with only 24 species. And it is not surprising that the members of Rubiaceae and Lauraceae families are dominant because common in that reserve because the forests are disturbed and located on highland, while only 34 species of Dipterocarps recorded from the reserve. For the endemism of the 821 taxa that have been recorded, there are 171 taxa that are endemic to Borneo and 25 taxa endemic to Sabah. And from our study, there is no plant species that is endemic to Mensalong Forest Reserve. But I would like to mention here that at least 2 new species of airweed were discovered, and I believe Dr. Kartini will share and elaborate more on this in her presentation later, whether those species are confined to the area or also found in other places. And this table shows the number of 13 threatened species in Mensalong Forest Reserve. 3 threat categories, namely critically endangered, endangered, and vulnerable, are categorized as threatened under the IUCN list categories and criteria. So there are 24 threatened plant species recorded from the reserve, of which 12 are listed as vulnerable, 9 endangered, and 3 as critically endangered. And the regazettement of the reserve into Class 1 protection forest reserve is timely and appropriate, as those critically endangered are now protected at least in this TPA. This slide shows the number of plant species in Mensalong Forest Reserve that are protected by state and international laws. And there are 63 species are considered as prohibited under the Sabah Forest Enactment 1968, 27 species protected by the Sabah Wildlife Conservation Enactment 1997, and 10 species are listed under CITES Appendix 2. So altogether, a total of 103 plant species recorded from the reserve that are protected by laws from felling. For the recommendation, we propose 4 high conservation value plant species, namely gaharu or Aquilaria bakeriana, serai kuning jantan or serai pulfolia, serai kuning gajah or serai gibusa, and serai majau or serai johorensis to be monitored. And the justification for the recommendation are these 3 species of seraias are critically endangered, as I mentioned just now in the previous slide. While the gaharu or Aquilaria bakeriana, though only vulnerable, but it is frequently harvested illegally for high market demand. So it is highly recommended to emphasize the protection of the reserve from any kind of wrongdoing, such as poaching, illegal harvest of plants, and illegal encroachment of the forest reserve in the conservation area management plan that will be developed later, formalized later. So in conclusion, this study recorded 821 plant taxa from the reserve, which is considered relatively high compared to other forest reserve in Sabah. And secondly, forests in Mensalong Forest Reserve contain high conservation value plant species, such as I mentioned just now, endemic, threatened, and legally protected species that requires attention as recommended just now. And all these data are very useful as a basis for the formulation of forest management plan or conservation area management plan of the reserve and surrounding area. So I would like to acknowledge the Chief Conservator of Forests, Deputy Chief Conservator of Forest R&D and PM of the Forest Department, DFO Kota Merdu, and of course the study team members for the high commitment in the field. So thank you very much.
Thank you, General John, for the informative presentation. Indeed, plants are pure exuberant species. Many of them are common, but some of them are on threats also. Well, if you have any question to Mr. John Sugao, please use the Q&A button. Click on it and start cranking your question. I see there's 132 participants have registered. Welcome to the Mensalong Forest Reserve Heart of Borneo Scientific Expedition webinar 2020. Now we're moving on to the next presentation. Do you want to know what's terrifying stuff about fungi? Fungi are the interface organisms between life and death. With delight, I would like to invite Ms. Viviane Paul to deliver the macrofungi of Mensalong Forest Reserve. Hello, Angel. The line is yours.
Thank you, Dr. Roman, for the kind introductions. You're definitely one of the fun guy here. Okay, so good morning, everyone. This is my turn to present about macrofungi of Mensalong Forest Reserve. So I just want to introduce my partner in crime that time. Thank you to my ex-intern, Bella Belket, and of course, my partner in crime, Dr. Jaya Silan. You are here, right? Together with his students, Puiling and Nalina. So this is the introduction, but I won't explain further because our keynote speaker already explained about the study area. So my focus will be on macrofungi. So what is macrofungi? So macrofungi can be divided into two phylum, which is Basidiomycota and Escomycota. So Basidiomycota is the club fungi and Escomycota is the sect fungi. So I won't explain further on that. But I just want all of you to get to know what is actually macrofungi. As you can see here, it has fruiting bodies that are too large to be seen through the naked eye and can be captured by hand. So you can see there has high variance in cap and stalk height, color, texture, and form. So the taxonomic group of the fungi, as mentioned by our keynote speaker just now, consists of eel fungi, jelly fungi, coral fungi, bracket fungi, and puffball. So the substrates, of course, you can see the fungi, the soil, dead woods, branches, logs, and on living trees. So of course, their roles in the forest, they are the decomposers, and they have medicinal values, and they provide foods for the animals, insects, and of course, they're symbionts, that is mycorrhiza, and their diversity is approximately 1.5 million. So these are the three objectives. First is to document the fungal diversity of Mensalong Forest Reserve. And my second objective is to generate a checklist for the Mensalong Forest Reserve Kota Marudu, Sabah. And the third objective, of course, it will act as an important knowledge for prospective researcher involved in researching the same genus or organism of the fungi. So these are the methods involved for the fungal survey. So first is the field survey. We went to the forest and of course we have to collect the fungi. So we have to do we did this study by using the opportunities sampling method, in which we have to walk through the area and collect the conspicuous specimen. So, and at the same time, of course, we also need to record the latitude, longitude, and the altitude by using GPS. And second, it will be followed by morphological identification, in which all the macromorphological characters were described based on the fresh specimen. Of course, photographs are needed here. And then when it reached to species level identification, right, the unknown species will be inferred using molecular methods in the later part. And third is the media preparations. So these are the photos of the media. Okay, and we will photograph the top of the media as well. But this one usually prepared by the interns. Okay, now we're reaching the result and discussion part. So as you can see here, this is the percentage of fungal family in Mensalong Forest Reserve, Kota Marudu, Sabah. So I will explain the top 4 dominant families for our finding. The first one is Polyporaceae. It dominates the most, which is 40%, followed by Ganodermataceae, which is 11%, and then Agaricaceae 8%, and Auriculariaceae 7%. So in total, we spotted 24 families of fungi, 33 genus, and 91 specimens of, and 91 species of species has been collected. So more than 80% fungal collection was spotted mainly from the substrate, dead branches, dead logs, and even leaf litter. So majority of the fungi specimen were found at an elevation of 700 meter. The elevation of lower and higher than 700 meter has less fungi. So previous studies have shown that fungi diversity is negatively correlated to elevation. So these are the studies conducted by the previous researcher. I already cite them in. Okay, now this is the number of fungi collected, as you can see here. Okay, like I said, this is the most abundant family, which is Polyporaceae. So you can see Microporus xanthopus, Microporus affinis, Traminus versicolor. And then this one is Panus bulbus. It looks like the wine cup here. Actually, it is a lentinoid fungi. And Corner 1981 described this species before in 1964 in Sabah. So Panus bulbus distributed mostly in tropical to subtropical forests. And it also provides microhabitat for small frogs. So when young, the fungi is purple in color. As age, they turn dark brown with hairy stripes. Now, the second most dominant species is Ganodermataceae. The substrate, of course, you can spot them around the dead logs or living tree trunks. They totally have these solid fruiting bodies, and they are the plant pathogens and can cause a white rot in living trees. And of course, they have antimicrobial properties against bacteria and some fungi. And one of the fungi, right, as you can see here, I'll use my laser. This is Amauroderma species, or known as Cendawan sawan, where it can treat epilepsy. Okay. Thirdly is the family Agaricaceae, in which you can mostly find them around the forest floor. So Lacaria species is ectomycorrhizal, and both of them are important in nutrient cycling. The fourth one is the family of Auriculariaceae. This one, if I'm not mistaken, dalam BM dia kulat korong, the common name. It's totally like macam jelly, and it can be eaten. Selalunya dia kena masak bersama-sama sup lah. Okay, and mostly you can find them around the twigs. The substrate is twigs. Okay, the other family spotted, including this is Podosecula species, look like pagoda. And then Cimatoderma elegans, Lacaria, and Gastro, family Gastraceae. This is the earth star fungi. And then Hygrocybe species, they usually have this bright yellowish to reddish in color. And then we have Ramaria species. It's totally look like squirrel. And then Hynum, Amauroderma, this one species, because a little bit difficult to identify. We need molecular study for this. And then Marasmius species, and then Mycena. You can say this is a glow-in-the-dark fungi. So this is cup fungi. I think most of you have spotted this in the forest. This is from family Sarcosifaceae, and got two differences for this coquina, in which you can see with hair, and then without hair. So commonly known as cup fungi, and then Coquina sulcipes. This one is usually larger in cup size, and they have shorter hairs at the cup edge, as you can see here. This one got hairy one. So the substrate is the dead woods. And then this one is the family Amanitaceae, known as Amanita species. Genus is Amanita. So this is based on Dr. Jaya communication with me. This is a new record for Sabah, Amanita CF cockery, and it has juno carpet development. And of course, this is mycorrhizal species, and it's usually poisonous. And this is the young stage of Amanita. Okay, now we already reached this family Gompaceae. Ramaria species is usually looks like coral, and I know they are totally looks like alley cat's hair, right? Can you see the resemblance? Yeah, alley cat's hair. Okay, and then many of you may spotted this if you are aware of this, but this is not fungi. Slime mold is, they are classified in the kingdom of Protista, despite many years of having been classified as fungi in the class Mysomyces. And then here comes the interesting part, family of Ophiocordycipitaceae, which is zombie fungi. And then this is, the photo shows that this is in asexual stage, and then they are known as Hirsutella species, and also first recorded in Sabah. And then for zombie fungi, how to identify them is usually fungi and hosts are totally specific for this study. So this is insect parasitizing fungi is not well studied in Malaysia. And then the first comprehensive study was conducted by Dr. Jaya ex-students before, which is Hafiz, and this is unpublished one. And then comprehensive molecular work are being currently conducted by Dr. Jaya's mycology lab members. So these are the selected references. I won't talk much about it. So as my conclusion, the preliminary study work of fungi will undoubtedly be available to those interested in studying fungi of Mensalong Forest Reserve. So it is hoped that this work will act as a catalyst to increase and promote more study to be carried out regarding fungi, especially in Sabah. So I would like to thank all the members that helping out in collecting the fungi, and of course, thanks to Dr. Jaya. So I already done with my presentation. Thanks for watching. Yeah, Dr. Ruben, are you here?
Vivian, for the informative mushroom presentation. Hello.
Okay, can you hear me?
Yeah, I can hear. Can you hear me?
Yes.
Hello.
Loud and
Okay.
clear.
I see internet connection unstable. Anyway, thank you so much, Miss Vivian, for the informative mushroom presentation. Indeed, all mushrooms are edible, they say, but some can be only eaten once in a lifetime because it's poisonous and you're dead. Okay. If you have any question to Miss Vivian, don't be shy. I know she's cute. Please use the Q&A button. Okay, moving on to our next presentation. Success doesn't just come and find you. You have to go out and get it. With great pleasure, I would like to invite the fourth presenter, Associate Professor Dr. Kartini Saibe, botanist and plant taxonomist from UMS, to deliver her presentation. Hello, Dr. Kartini. The line is yours. Dr. Kartini? Hello? Dr. Kartini? Hello, Dr. Kartini? Yes.
Is offline.
Oh, really? Okay, maybe we could move to the next presentation. Land is not merely soil, it is a fountain of energy flowing through a circuit of soils, plants, and animals. With great pleasure, I would like to invite the fifth presenter, Madam Louisa Majoaquim, a botanist and ecologist, to deliver her presentation. Hello, Louis. The line is yours. Unmute. Can you unmute yourself?
Sorry. Okay. Thank you for the kind introduction. I'm going to share my slides now. Okay, very good morning, everyone. Okay, so I'm going to present my study, which is on determination of total organic carbon in soils of Mensalong Forest Reserve. And this is a collaboration study with the soil survey team from SFD during the Mensalong expedition. So I'm going only to present a part of my finding, which is on total organic carbon in soils. For my introduction, most soil carbon is found in organic form, which is contained in soil organic matter, what we usually refer to as humus. And organic carbon consists of a wide range of molecules ranging from simple acids and sugars to complex humic substances with large molecular weights. It is also a product of decomposition of litter and humus, but it may also originate from the exudates of plant roots. So there are many functions for this organic carbon. So it plays an important role in below ground processes such as maintaining soil functions, and this includes decomposition and also in nutrient cycling. And soil carbon also plays an important role in maintaining soil fertility and in sequestering carbon. And all of these, of course, contribute to the health of the soil. And in addition, organic carbon also contributes to ecosystem services, such as for food production through the maintenance of soil fertility, and also contributes to the regulation of global climate by greenhouse gases mitigation. And the objective for this study is to investigate soil TOC across different forest types and soil depth. Okay, moving on to materials and methods. Soil sampling was carried out in 8 forest plots with elevation ranging from 450 to 900 meter above sea level. And these 8 forest plots are identified as lowland mixed deep rock cut forest, LMDF. And I would like to rectify here that there is no low mountain forest plots have been established. So the other plots are all being established in the upland mixed deep rock cut forest. And LMDF plots have been established at 450 to 580 meter above sea level, and the upland forest has been established at 610 to 745 meter above sea level. So soil samples were collected using soil core sampler in 5 rectangles for each depth for each plot. And soil sampling, once it's been done, all the soil samples were composited for each depth for each forest plots. So this gives a total of 24 samples. There are 3 plots in the lowland and upland, 3 plots in the lowland and 5 plots in the upland forest. So lower mountain forest has been grouped together as upland forest. Now for soil analysis, we carried out TOC and also pH determination. So TOC was extracted using 0.5 molar potassium sulfate and then analyzed for TOC on Shiatmatsu TOC TN analyzer, while pH was determined using the slurry method. Okay, moving on to results and discussion. So this diagram here shows pH value, and the y-axis represents pH, while the y-axis represents increasing elevation and increasing soil depth. So there is a decreasing, generally a decreasing pattern through depth, but the results is invariable. probably due to the narrower range of soil depth. And lowland forest has a slightly higher soil pH compared to the higher elevation forest. Next, for total organic carbon, the same diagram is shown here, and instead of pH, the y-axis represents total organic carbon in milligram per liter, and the x-axis represents the same parameter, increasing elevation and increasing soil depth. So as with pH, there is a increasing pattern through depth, but it is the same as soil pH. And in addition, higher elevation forests, the upland forests, they have higher soil TOC compared to the lowland forests. So previous studies have shown that soil carbon increases toward higher elevation, and higher elevation forests could be severely impacted in the event of forest clearing or deforestation. Due to higher accumulation of organic carbon in the form of humus, soil carbon loss could be higher in these forests, and slower organic matter turnover at higher elevation forests could potentially reduce soil organic carbon content in these forests. So in conclusion, this study has implication on the management of soil organic carbon in the form of soil organic matter in degraded ecosystems for the prevention of potential soil carbon loss and nutrients from the disturbed ecosystem and forests with low nutrient availability, such as Karangas forest. And as part of forest reforestation measures, post-deforestation treatment of soil carbon in degraded forest should be carried out to assist in the recovery of soil carbon. And this can help to restore soil fertility, especially in degraded forests. And with that, I can talk. Thank you for your attention, and I give it back to Dr. Ruben. Thank you.
Thank you, Madam Louisa, for sharing your findings. To protect the soil, you must have a lot of vegetation cover. If you have any question for Louisa, please use the Q&A button and ask question anything about organic carbon. So moving on to our next presenter. Franklin Roosevelt used to say, "The nation that destroys its soil destroys itself." With pleasure, I would like to invite the sixth presenter, Miss Esther. Come on, give it a smile. Our soil scientist to deliver soil and above ground carbon of Mensalong Forest Reserve. Hello, Esther. Please grab the line. Unmute yourself. Esther, please. Esther, can you unmute your microphone?
Okay, hello, sorry. Can you hear me?
Yes, loud and clear.
Loud and clear, thank you. My boss and Aka, my sifu. Okay, today I'm going to present a preliminary study of forest ecosystem services towards soil and forest conservation at Mensalong Forest Reserve. And this study is co-authored by me, Mr. Alex, Dr. Rubens, Mr. Jupiri, Bos John Sugao, Amirul Faiz, Sabini, Madam Louisa, and Johnny. So I think I shall proceed my presentations. Okay, for the introductions, forest ecosystem services can be defined as benefits from our valuable forests to support human being that includes economic, social, and environment. So in this study, we're focusing on forests and soil and as important ecosystem provider So forest and forest soil play a complex role and intrinsically linked. Soil is formed by a continual interactions between soil system, biotic and topography, and it's very important to supply air, water, nutrients, and mechanical support to sustenance plants, receive and process rainfall, and active ongoing exchange of gases between soil and environment. So as we all know that the importance of forests is to generate income through timber industry and then provide job opportunity. Okay, and then forests act as an habitat for biodiversity and livelihood for humans, and then a key to mitigate climate change where forests absorb harmful greenhouse gases, and then for watershed protections, where forests regulate water cycle by lowering surface runoff, recharge groundwater, and continuous yielding to rivers. And then forests also important to prevent soil erosions, and at the same time to improve soil quality. So the aim of this study is to investigate the significant value of forests ecosystem services, towards soil and forest conservations in promoting climate change mitigations. So the scope of research includes investigations of soil characteristics such as parent material, their drainage condition, soil depth, color, textures, and as well as slope gradient. So we did include estimations of above ground biomass carbons and its potential carbon gain, and some recommendations activity that can support soil and forest management at Mensalong Forest Reserve. So the methodology for field soil assessment, a total of 8 soil assessment points were conducted throughout the expeditions. We examine to examine soil depth, color, texture, and site descriptions. And we did also collect sample from depth 0 to 15 cm and 15 to 30 cm for lab analysis. And then all the GPS coordinate of all sampling points were recorded. Okay, and the second methodology, estimations of forest carbon study. So here we're using CAO, Carnegie Airborne Observatory spatial data to estimate average carbon density for this reserve via statistical modeling. So this is how we conduct our field soil assessment. Okay, we did our soil sampling using soil auger, and then we arrange the collected soil in a horizontal position so that we can examine the depth easily, is examine the soil color using Mensalong chart, and then examine the soil texture. So as the result, soil at Mensalong Forest Reserve was classified as Crocker Association with dominant unit, soil unit of the Cresol district and chromic cambisol and lithosol. They are parent materials from sandstone and mudstone with well-drained conditions, topography in steep terrain, and the elevations of soil assessments varies from 465 to 925 meter above sea level. So this is the result of soil assessment. Soil colors varies from dark brown to yellow, and textures from loamy sand to sandy clay loam. Okay, and these were the pictures of soil profile from for soil assessment point 1 to 4. Soil color in brown, which indicate a well-drained soil. And then this goes to soil assessment point 5 to 8. Okay, okay. This is the slope map of Mensalong Forest Reserve. Okay, slope was grouped into 6 classes, which from 0 to more than 25 degree. Slope classes with more than 25 degree covered about 26% of the total area, which is the highest. And then the lowest goes to slope classes with 0 to 5 degree, which covered about 9% of the area. And another 65% covered by 4 slope classes, which stated in the map. And then we did also forest quality stratification map for this reserve, and it's turned out of five stratums of forest quality, which indicates from stratum 1, good forest, down to stratum 5, small crown to bare area, and then stratum 3, which indicate as poor forest, covered about 42% of the total area, and the lowest covered by stratum 5 with about 2% or 152.58 hectare. And this is the CAO above ground biomass carbon density maps. Okay, and the highest carbon density stated in dark green color. So it can be seen very clearly around here. Okay, with more than 200 ton carbon per hectare density. Okay, and then the lowest average carbon density shown in red color, which is mainly around here. Okay, the average carbon density less than 15 carbon ton per hectare. Okay, so for the estimations of above ground biomass carbon at Mensalong Forest Reserve, the average carbon density was at about 138.23 ton carbon per hectare, and potentially can get up to maximum of 377 ton carbon per hectare. So the current total carbons for Mensalong about 1.1 million tons, and the current carbon dioxide equivalent about 4.04 million tons, and the potential carbon gain in total in future for Mensalong around 1.9 million tons. Okay, let's move on to discussion part. As I mentioned just now, soil in this reserve was classified as crocker associations, which form on mountainous terrain, occurs on some steepest land with elevations more than 330 meters, very high, narrow ridge, and long slope with angles generally greater than 25 degrees. Slopes are rocky and prone to landslip. Valley are narrow and boulder stream. River are fast flowing, generally not very deeply weathered, and shallow to moderate deep and highly erodible. Yeah. So like other forest reserve, like Mensalong Forest Reserve have the same important value as a global carbon cycle in mitigating climate change, where forests have the ability to sequester and store carbon. Trees absorb carbon dioxide and then release oxygen during the process of photosynthesis. Thus, it helps maintaining the temperatures and oxygen level of the atmosphere. So if assumptions, pristine forest as reference for Mensalong Forest Reserve, the average carbon density potentially can up to maximum of 377 ton carbon per hectare, or 1.9 million ton in total. So this average carbon value, potential carbon value, is higher than current average carbon density reported at Maleo Basin and Inbak Canyon, with their current carbon density 220 and 228 ton carbon per hectare. So if we have that opportunity to enter carbon market in future, with Mensalong Forest Reserve as reference, assuming 35 years of recovery with low and high ton carbon dioxide equivalent prices in USD, $3 to $50, and the estimated income from carbon market around Ringgit Malaysia 2.8 millions to RM 12.3 millions per year. Okay, this is just an assumption. Okay, and then Mensalong Forest Reserve prone to threats due to an uncontrolled human activities, with series of conventional logging in the past and wildlife poaching, and prone to forest fire. So the impact or implication of these threats contributed to soil erosions and compactions, which degrade soil physical properties and nutrient loss, and will directly decrease productivity of forest vegetations. Secondly, degradations of watershed, where soil erosions may aggravate the accumulations of sediment in the river, which can reduce water quality, and this will affect the clean water supply for the adjacent community livelihood, especially communities who lives and fully depends on water from Sungai Paimaitan to Sungai Bongon, Sungai Mensalong to Sungai Kepuakan, and down to Sungai Sugut. And it's also affecting the freshwater ecosystem services and habitat loss. Soil erosions due to humans' activity in the past, steep and rocky terrain of this reserve, and unpredictable geological weathering will increase the possibility of landslide happens in Mensalong. Okay, there are recommendations for forest and soil management for this reserve, which include tree planting to improve soil quality and at the same time reduce soil erosions, forest restorations and rehabilitations activity, regular patrolling, comprehensive forest fire prevention, especially during long drought, and then boundary demarcation. So as the conclusions, forests and soil are important service provider and importance to generate income, global cycle, prevent soil erosions, improve soil quality, watershed protections to supply clean water and home for biodiversity and human livelihood. So soil in Mensalong, again, classified as crocker associations, which occurs on steepest land. Soils were well drained. Colors varies from dark brown to yellow, and the textures luminescent to sandy clear loam. So the estimated average carbon density around 138.23 carbon ton per hectare, and potentially get up to maximum of 377 carbon ton per hectare. And in total, about 1.1 million tons, and their current equivalence carbon dioxide equivalents around 1.04 million tons. So forest restorations and habitations activities are highly recommended to continue the role of forest and its ecosystem as benefit service provider. So this is all from me. Thank you very much from me and my co-author. Thanks. Over to you, boss.
Sorry, I forgot to unmute. Thank you, Miss Esther, for relaying us the value of the forest ecosystem for the benefit of the human well-being juga kan?
Yeah.
Anyway, if you have any burning question about these studies, please use the Q&A tab down below. There's announcements. Dr. Kartini having difficulty presenting her paper because there's no power in her place. So her slot will be rescheduled to the afternoon session. And after Hubert Pitol's talk, one presentation from the afternoon sessions, which is the study on endurance by Panglima Muhammad Salan, will fill in her slot. Okay, moving on to the next presentation. Birds are the indicators of the environment. If they are in trouble, we know we will be in trouble too. So let me invite you, Mr. Hubert Pitol, our ecologist, EE practitioner, and avid birders, to deliver bird survey in Mensalong Forest Reserve. Hello, Pag. Catch the line.
Hi, Pag. Can you hear me, Pag?
Loud and clear.
Okay, thank you, Dr. Ruben, for the introduction. Okay, I'm sharing my slide now. Okay, can everyone see my slide? Yeah, okay, all right. So I'll talk about the survey that we did in Mensalong Forest Reserve on birds. Okay, why we did the survey was because we just want to know about the avian diversity in Mensalong Forest Reserve, all the common things like species richness, species list, and abundance index, and all the other things that we need. And also, how food resources are utilized by the birds. Okay, for Mensalong Forest Reserve, the survey sites, you can see here, this is where we went to, and like it's interesting to know that these are the places that we went to, which also correspond to what Ms. Esther said just now, which are the best parts of the forest, except for site number 4, which we tried to go towards the lower montane forest. Okay, next is our methods are very similar and normal methods for rapid assessment, which we call the McKinnon-List method, although we have a bit of adjustment to our 15 species list method. We use abundance data as well. So the survey days are around 4 days, approximately 4 hours per day, and including evening as well. And we detect the birds visually and also orally. Observations were conducted along old trails and old logging roads, or skid trails, more accurately. Okay, the more important things for us using the McKinnon-List method is because we have abundance data as well within the list, we really have to prevent double counting of individuals, and because they're in the upper MDF forest, we have to check twice, and also compare it with the books, and also listen very carefully, because we are unfamiliar with the calls of the birds in that area. Okay, our survey schedules are as follows. Only on the 4th day, 3rd August, the 4th day, we started late because it's quite far away from our base camp. So that also affects the how many birds we can see. The detectability of birds go down after 7, 8 or 9. Okay, let's go straight to the results. We found we had 23 lists. Each list is 15 species. Altogether, we have 566 individuals. All in all, 101 species of birds, and within 37 families. And the Shannon Diversity Index is quite high. It's quite diverse. According to that level, 4.1 is actually quite high, because normally it's around 3.5. So, and the evenness is 0.7, is also quite even, meaning to say all the birds have similar numbers of individuals. As a side note, the Sabah Park staff who also went for the expedition, they detected 103 species from 37 families during the expedition. Okay, for this is the list of Bornean endemics and their categories under the IUCN Red List. So you can see only the black-throated wren warbler and the Bornean tree pie were categorized as near threatened, and Kinabalu serpent eagle and the Bornean necklace partridge are as vulnerable. Okay. Okay, and just for the whole list, all the species listed as near threatened and vulnerable, these are the list here. You can see around 24 species are near threatened, and 4 species are listed as vulnerable. Although there are some that I don't agree with this, but this is how it is. There are some that are very, very common in all the places that we've been to. Okay, these are the top 10 species with the highest number of individuals detected. Of course, they also these are also shared rankings. You can see the gold whiskered barbet, orange-bellied flowerpecker, and so on. They're very high numbers of individuals detected, but These most of these species are mainly detected by the calls, meaning to say that they have very high oral detectability. It doesn't mean that other species are not there. It's just that when the forest is thick, we may not be able to detect it visually, but we can hear. But those that are very non-vocal, we cannot detect them. So, it's difficult to detect them. So this rank, for me, I don't really, we cannot compare from one between species in different areas, the relative abundance. However, when you just look at it from all the species, it's good to compare Mensalong. You can see Mensalong here is the thick line. This is all the places that we've been to. So generally, the rank abundance curve from all these forest reserves, they're very similar in terms of how abundant they are. Generally, all this one. So as you can see, they're quite similar in all the 7 forest reserves. Of course, the length of the lines depends. That is the number of species, but the curve is very similar. So yeah. Okay, the top 5 most specious family, meaning to say the top 5 species families which has the most species. Of course, they're the bulbuls with 13 species, and the sunbirds, spider hunters, together with the babblers, they both share 8, and then the rest goes down. These are the most common, and you can also see that most of them are also detectable by their calls. This is the 10 families with the highest number of individuals detected. So the bulbuls, they have it's 86 individuals. It goes down with the rest of the families. You can see that. So that means most of if you see the top 3, almost all the birds come from these 3 families, the top 3 families. Okay, this something that we can do from the McAnally's method is to get a species accumulation curve. As you can see, we at 101, 101 species, the curve has not achieved asymptote yet. It's not level yet, so there's still more species to be found or to be detected. However, for us doing rapid bird assessment, like 3 or 4 days, we use this to see it. And also another one is we estimate the species richness using the super duplicates online calculator developed by Chow et al. It only requires the total number of species observed, which is 101, and the total number of species observed only once. That is why in our method for McAnally's method, we have another column for abundance data. So I think for 3 or 4 days research, we get a more accurate estimation of the species richness using these super duplicates, mainly because of the number of species observed only once. Okay, 3 more minutes. Estimated species richness is around 118 species. So we did not detect 17 species according to this estimation. Okay, now we go to the species in their natural habitat and feeding guilds. As you can see from this graph, this diagram, a lot of species live in the forest or forest-dependent species. So there are 93 species that are forest dependent, although 74% are very strict and 11% are outside the forest. And then on their feeding yields, there are 57 species of insectivores, which are the most, and also 48 species which are strictly strict insectivores. Other insectivore others are a mixture of frugivores and insectivores, but 48 species are strict. And there are also frugivores, of which 29 species are very strict. This is to say that the forests still have a lot of resources for in terms of insects and also fruits. Okay, we come to some bit of discussion. The diversity can still be considered high, taking into account the low observation hours of about 20.5 hours. We can see that there are mountain elements within our species list, something like the Bornean tree-pie, Bornean iban, Everest white-eye, spotted fantail, chestnut crested euhina is very common, and the rest. Okay, out of the 10 endemics, 4 species are only found in upland and mountain forests. The high number of forest and forest-dependent species reflected the relatively intact forest ecological functions, at least at our sites. Yeah, and with the insectivores, 57 species from 28 families, it shows that the insects were an abundant food resource, and also correct me if I'm wrong, maybe there's also a high number of flowering trees because of the high number of frugivores as well. So in conclusion, I think the first ecology where we went to, I couldn't say that for the whole of the forest reserve, but the sites that we went to, they were quite stable forest ecology. That quite stable ecology. So I would like to thank the team, Cecilia, Vivian, and Lisa, all those involved in organizing the wonderful expedition, and also the others who for the active participation and sharing of the information during the survey. With that, thank you very much. Over to you, Pag.
Okay, thank you, Hubert. That's a wonderful information. At first, I see the forest quite depressing. Esther showing some degraded forest, but then you said the birds still happily. That's very assuring. Thank you, Pak.
Thank you.
If you have any question on Hubert's presentation, please fill in the question and answer window. I would like to ask the Secretariat, is Dr. Kartini online? Hello, Beatrice.
Online. Hello.
Yes.
Yeah.
Hello. Yeah. Yes, I am.
Is Dr. Kartini online?
Yeah, Dr.
I
Kartini
am.
is on the line.
Is on the line. Can she present? Right?
Yeah.
I think can Dr. Kartini because of your connection problem, right?
Yeah. There's power blackout, I guess.
Yeah.
I am ready now. The power is on.
All right. Okay, you're back. Okay, so
We cannot see your face, Dr. Kartini. Is
Yeah.
your connection okay?
See, I already on my video. Oh, I see. Okay, let me choose. Choose. Select a camera. Background.
for your information, there's a lot of question has been forward in our Q&A. That shows a very good response. And I thank you for the speakers who have answered them directly. And but let the question flows in, a lot of them. The more, the better.
Yeah, Doctor, I got one question to answer live later on for
Okay.
Mr. Robert Mijo.
Keep it. I know you're excited. I'm also excited. I got one question, but I cannot ask question, right? Never mind.
Yeah, you definitely
Since you
cannot
all,
ask question.
yeah, since you're all in the center, I can bother you guys later.
Yeah, do you need help, Dr. Katini?
Why I cannot, yeah, why my picture not there? Why?
Are you covering your camera, is it?
Oh, yes. Sorry. Yes.
Okay.
Okay, now we can see your beautiful face now.
Yeah, over to you, Dr. Ruben.
Okay. Dr. Kartini is back. So
Can I do something with the background first? I'm sorry. No.
I think your background is okay, Doctor. Yeah.
All right.
No
Okay,
worries.
sorry for
Yeah.
everything. Okay. I think that's all right. So just can we go on?
Yeah, maybe you
Will
want
I share
to
my screen?
inform the title? Yeah. Yeah, sure.
Thank you. Can you hear me clearly?
yes, we hear you loud and clear.
All right. I am very sorry for all that trouble. I think that is one of the, among others, the interruption that we will have to face when you work from home. Okay, the title of my presentation today, thank you for having me, Preliminary Survey of Arasia of Mensalong Forest Reserve, Kota Marudu. Okay, even though Arasia, we have a lot of leaf morphological shape, but then it can be easily distinguished by its inflorescence, which is consists of the spathe and spadix. So this is the spadix, and this is the spathe. This is the bisexual flower, which is mostly in climbers, and this is the unisexual flower when you can see the female and the male zone. So the Araceae comprising about 7 subfamily, 121 genera, and 6,000 species, and mostly found in tropical area. In Borneo, we have about 433 formally described, and in Sabah, we have less than 100 that have been described. So that is very important if we have this kind of scientific expeditions so that we can increase our knowledge in the current status of Araceae in Sabah. So preliminary survey on Araceae in Mensalong was conducted on 30th and 31st July for 2 days. 2 elevations were surveyed, which is 750 and 500 meter above sea level. The entrance on the first site was from the main road, and the second site from small plantations. And both sites located about 2 kilometers from the starting point of the forest boundary. So all the Araceae found were recorded. Several specimens were collected, grown in nursery, and observed the flower characteristics. Identification was based on Wong 2016, Boyce 2004, Mayer 1997, He and Yuzami 2001, and personal communication with Peter Boyce. So we recorded 3 subfamily and 6 genera with 12 species of Araceae in Mensalong Forest Reserve. So this is the subfamily, Potoideae and Monstroideae is the climbers. Both, all the species, Photoscandens, Scindapsus longistiptatus, Scindapsus species found on both trail. The subfamily Oroideae, Aglonema nitidum, Alocasia wongiae, Homolamina species 1, Homolamina species 2, Homolamina species 3, Cismatoglottis asperata complex, Cismatoglottis propex, Cismatoglottis species Calyptroclade, Cismatoglottis trifasciata complex. I'm sure we can find more than all these species if I have taken more time and more area to be surveyed. This is the Homolamina species. Homolamina is a aromatic herbs, can easily distinguish by its persistence path. This is the Homolamina species 1, found in 500-meter trail. And these two species, species 2 and species 3 of Homolamina, found in the 750-meter trail. This is Cismatoglottis propex, first described in 2019. The type specimen is from community land, the Kion Som waterfall recreational park, and Inanam. So it's just nearby Kota Kinabalu. And this is the Cismatoglottis. I'm not sure what species is that, but it is from Calyptroclade. This is Cismatoglottis asperata complex. We found it complete with inflorescence on the first day. So this is actually ready to be described. So on the first day we found this, and it's really take my mind off from the expedition. All I want to do is to go back because the inflorescence need to be preserved. This is the Cismatoglottis. We collected it in sterile living specimen, grown it in nursery, and the inflorescence characteristic shows that it's a novel species from Trifasciata complex. This is how it's very important to have the sterile specimen collected and grown in nursery, because the inflorescence actually the main characteristic when we describe, I think, any plants, not only Araceae. So the asperata complex, the Cismatoglottis zynudii, and the Trifasciata complex, the Cismatoglottis schaffii, were published in 2017 as the first record in Sabah, and the type specimens of both complex are from Tawau Hill, Sabah. And this is the climbers. These are the climbers that found in all the trail. This Scindapsus species is a huge climber. It's very common, but never been described because we haven't found the inflorescence yet. And this is the common Photoscandon and Scindapsus longistiptatus. So Mensalong Forest Reserve actually accommodates many interesting species. And even though we have a slight differences in the distributions of Araceae in both elevations, but actually it's very hard to make significant conclusions because the time frame was short and the area covered was small. And I really recommended that this Mensalong Forest Reserve to be enforced as a protected area. So I would like to take this opportunity to acknowledge University of Leicester Sabah, Peter Boys, and Sabah Forestry departments. Thank you very much. So up to you, Dr. Ruben.
Oh, yeah. Thank you, Dr. Kartini, for the wonderful discovery. And now marks the time, which is 10:53. Now we are entered the question and answer session. So there'll be 30 minutes. So let's see what's in the bucket. Q&A. So there's quite a lot of question here.
Yeah, Dr. Ruben, before that, maybe we can ask all the speakers to please switch on their webcam.
All right, okay, that's a good idea.
Yeah, so that we can see the
please
faces.
turn on your microphone. Also, microphone? Is the microphone on?
Yeah.
Okay, so can we look at, okay, so most of them has been answered. Okay, let's see the first question. I think we go according to the first come first serve basis. So the first question came from Bernadette Jorman. Wow, so early, 9:42. Okay. Faiz, what was or were the past anthropogenic activities as you mentioned? I believe Faiz just didn't mention. I follow very close your presentation. So Faiz answer, the obvious one is the development of Kota Marudu Rano Main Road. Prior to that, the study site functioned as class 2 commercial forest, meaning that timber extraction activity is permitted. Fortunately, the site is reclassified to be class 1 protection forest reserve. So whatever I believe the disturbance would be activities from illegal, encroachment and poaching. So let me see. I hope that answers Ben's question. Okay, the second question is, any primate sighting in Mensalong Forest Reserve? Oh, this is very advanced question. This presentation on mammals finding is in paper 9, is in the afternoon session. If you can wait, you wait until the afternoon session. I think Faiz, also the same person, will answer this question. But I think Faiz has answered here, but I reserve that for the afternoon session. Move on. Bernadette has compliment Faiz question and willing to give assistance in the future. That's good. Nur Ain Aceh posed a question to the angel, Miss Vivian, regarding on the habitat for the macrofungi. Can it be found outside the protected area? What is the level of protection for macrofungi in Sabah at the moment? Can you answer that, Miss Vivian? I want to hear your voice. Miss Vivian?
Yeah, I'm here, Doctor. Hold on a second. Yeah, I need to trace the question first.
You have answer, but I find it a bit blurry.
I have answered it.
Yeah.
I have answered it.
I think it's better if you do it orally.
No, I already lost that question, so give me some time to trace the question first, Doctor, because there are so many questions.
Okay, we move on. All right, there's another question for you. What frog species was it that used the fungi?
This one it's quite a long name. I don't know whether I pronounce it correctly. Batrachochytrium detrobatitis. Or I call it as BD frog. The short name is BD.
Betis kan?
Yeah, and it's not prevalent in Malaysian stream. Maybe Sahlan know about this because I'm not the frog expert, but I only know this this frog is associated with that fungi.
Okay, we reserve that to the afternoon session then, to Pangeran Salan.
Yeah.
So next to the next question. Hi, Faiz. Alessandra here from SABC. It's quite alarming that encroachment poaching are still happening within the area. Was the picture taken during the expedition? Sorry, I missed the explanation on the picture. Based on Mr. John Sugao intro just now, 60% of the local communities still depend on the natural resources. Hope they don't get it directly from the forest reserve. Agree with the environmental awareness and maybe alternative livelihood introduction. And Faiz has answered this. All right. Okay, I think I would like to call Faiz to answer this.
Okay, boss.
Yeah.
Hello. Okay, yeah, the aerial image was taken during the expedition, but we are not sure whether the open burning occurred within or outside forest reserve. But what we sure is the open burning occurred in adjacent to the forest reserve, so the threat is real. We also observe some kind of encroachment, especially at the lower elevation of the reserve, which is next to the main road. But I didn't post the image in the slide. Yeah, that's all.
Okay, thank you, Faiz. Yeah, and perhaps this question also can be addressed in the evening session. Okay, I think there are two papers concerning about social. Okay. Can we move on to the next question? Vivian, have you found your question?
Regarding on the habitat for the microfungi, is it, can it be found outside of protected area? Okay.
Yeah.
This question is from Nur Ain Aceh. So hi Ain. Yeah, it still can be found outside of protected area, but species diversity of microfungi sometimes depends on the type of host around the soil type. So if you asking me about the level of protection in Sabah itself for fungi, it is still very low, and this definitely agree by Dr. Jaya as well. And this topic of fungi is actually under study because people tend to be ignored of these small species, and people not really care much about them. So hopefully we can recruit more fungi group in the future. Okay, that's all from me, Dr. Ruben.
Thank you, Vivian. I hope I'm not in the list. Okay, thank you.
You definitely one of them, Doctor. Fun guy.
I'm gonna move on. Wow, you so, man, look at it. Another question is about fungi. From Chia Ying Ling, for zombie fungi, I wonder, can we find them on growing on organic matters instead of parasitize on insects or worms? I ever discovered a fungi which resembles zombie fungi, but it grows on forest floor or on dead organic. And well, you answered that. Not many species, cordyceps and ophiocordyceps, needs a host insect. You want to answer this?
And then if Miss Ling from Forestry Department of Sarawak wants to know further about this fungi group, I mean this zombie fungi, Dr. Jaya's currently working on that. They do some molecular work on that. So this is not my field of expertise, but more or less I just know the basic thing. Yeah.
All right, thank you, Vivian. You really catch all the participants. You see another question about your study. Thank you for your presentation by Azad Jangil. Thank you for your presentation. Just wondering, how was the caterpillar fungi came in Sabah? Are they aliens?
This is I get this info from Dr. Jaya from our personal communications. Actually, we call it as zombie fungi, not caterpillar fungi, because it's endemic only to Tibet, China. Yeah. And then cordyceps is a genus of parasitic fungi that grows on the larvae of insect, for your information.
How they came in Sabah?
How they
I think that's the question.
Yeah. I think it's hard for me to question that. Yeah, I'm not sure, Doctor, how they come to Sabah.
Okay.
Yeah.
Okay. Terrorize and fungi, so it's fungarize. And it's okay. Anyway, hi boss Vivian. This is by Sasi Kuma Tangaraju. Oh man. During the collection of the fungi, does the fungi have the common characteristic of the habitat other than their hosts? For example, like what species of fungi usually found around what species of tree nearby? If this happens, is it related to mycorrhizal reaction between the tree and fungi or something else? That's very long.
Yeah.
Yeah, can you answer that?
If I already answered based on the Q&A section pen over there, but Sasi, if you want to have this long discussion with me, yeah, you can do so by using Google Meet, or you can contact me via WhatsApp. But this is what I can answer you briefly based on Q&A. But if you want to know further, you can always look for me. Yeah.
Okay. Okay, there's also a question by Robert Mijol. Thank you, Vivian, for interesting presentation. Any fungi in
Thank
Mensalong
you.
that can be used as bioindicator of forest health? Okay, this is the last question for you, Vivian. Okay.
Yeah, I know I'm the most. Okay, banyak questions hari ini. Thank you for your attention. I really appreciate that you start to focus on this fungi topic. So I will answer this live. Of course, for Lakara and other species can be indicator for the forest health. So if you asking me how the Lakaria species looks like, I will share my screen right now. Okay, can you see this one on top? This is the Lakaria species. This is one of them. You can indicate the forest health by looking at this species, Lakaria species. Okay, that's all from me, Doctor.
Okay, thank you, Vivian.
Mhm.
Okay, next question by Azad Jangil. Hi, Dr. Go. Oh, I don't know if anyone here by the name Dr. Go. I think the nearest one would be George. Huh? George Hubert Pito.
Yeah, I think it's
Because he's asking
I
about
think
birds.
GH
Dr.
because of the
Go.
GH.
Hi, Dr. Go. Okay, I read the question. May I know what is your point view for the bird species that are common in a particular area but have high rank in conservation status? Hubert?
Okay, I answered that lah, but I think what it means here in high rank conservation status is, I think the vulnerable and near threatened status, although they are just for common birds. But I think that it all boils down to this lack of baseline information on the abundance of this species. I think that's it. Um, we all know that a lot of research has been done, but mainly I think by the academic institutions. So how common the bird is now, um, we a lot of species we don't know. Like you see my slide just now, even the black and yellow broadbill is considered near threatened. Although there's a very common sound you hear in any lowland rainforest. I think, like I said just now, it all boils down to just lack of information. But the nice thing now is what people can do is go to eBird. If you're a birder, a lot of the birders here in Sabah have already contributed to it. Any place you go, you let's have a list, and then you contribute to the eBird, which automatically maps where you are according to your GPS, and so that I think if they link with IUCN, that is a more what do you call it, reflects the bird populations or abundances more accurately if more and more people do that. But that is the reason why forestry department is also or we are trying to come up with the rapid assessment methodology which only takes like 3-4 days, and then you already cover more than 80% of the species list of the species there. So so the more places we can go, the better, the faster. Yeah, it just takes a few days to get a reliable data. Of course, we modified the mechanized method a bit just to get higher abundance, more accurate abundance data because of the short times, short survey days. But yeah, what we're also trying to do is actually to promote this into. I've talked to the Borneo Bird Club, some members. We want to train them in doing bird surveys because it's part of citizen science. So the mechanized method is very simple. They don't have to analyze the data just like what I did, but it's very simple and people can get abundance data or species species list data faster and more reliable. I think that's all I need to say. Thank you.
Thank you, Hubert. Okay, there's one question I think John could answer this by Kalsum Yusa. Will there be some kind of protection act for Arasia now that it has becoming famous with hobbyists? A lot of forest areas can be seen for sale in social media nowadays. John, can you answer that?
Okay.
Hello, John.
Seems like Boss John got connection problem over there, is it?
Hello, John. Okay, I think we reserve that question. I think we move on. There's one more question by Mr. Anon, anonymous attendee. You mentioned that the forest is degraded. So, do you think that the birds already tolerant by the disturbance? I think this question for Mr. Hubert. Hubert, can you answer that?
Yeah, I mean, the from Esther's map just now, it was quite interesting, because we went to all the good forests. So that was where we went to lah. So I didn't know that there was so much degraded forest. Well, also I don't know, this Mr. Anonymous attendee is what they mean by degraded. You know, for us, if we are into forest ecology, we may have different meanings of degraded forest lah. However, trying to answer your question, I did not go to the degraded forest on the first, second, and third day. Went to the fourth day, which under Esther's map was supposed to be degraded, but it is maybe degraded lower montane forest. But as a forest, as a layman punya thing kan, the forest was still quite okay, and the birds there were still very good lah. However, like you, if you saw in my one of my presentation, the graphs, they are, when you lose forests, there are many species that will be gone. I mean, will move out, or for that area they will be gone, because they are highly dependent on forest sejala, only a primary forest. So it will definitely affect, have a negative effect to the birds if it's degraded. Definitely the bird species count will go down, and other families who are which are more open area birds, forest age birds, those are the ones that you can see there. Yeah, I think that will answer it. Thank you.
Thank you, Hubert. Thank you. Right. Sorry. Yeah. John is still not back on the line, so.
Okay, I'm on the line.
Oh, you're on the line. Oh yeah.
Yes.
Back.
Okay.
I thought I lost you. Okay.
Can I proceed?
Yes, you go. I give you 1 minute only, man. You just okay. Since you're a nice guy.
Okay. So very fast. We cannot list the Arasia under the any act or protected species because it is not threatened so far. For example, it's not under IUCN Red List, or not under the Wildlife Conservation Enactment, not in the Forestry Enactment. So we cannot simply put it. But for sure, under the Forest Enactment, we are not allowed to enter any forest reserve. So that is the first thing that we have now. So I think it will answer the question for the time being. We cannot enter the forest reserve to encroach and collect the specimen from any forest reserve. Actually, the act, I mean the enforcement, I mean the regulation is there. Thank you.
Thank you, John, for the answer. Yeah, I think we have gone beyond the question and answers session about 4 minutes. So I think it's time to wrap it up. So with that, I have to close this Q&A session. Thank you for all the constructive question and answer that you all have been put forward. You guys are truly great and fantastic. So this is the end of the morning session. So thank you for allowing me to facilitate this morning session. Now I'm handing over this platform to the webinar committee. Bye.
Okay, so thank you again to Dr. Ruben for being such a great facilitator for this morning and very, very fun facilitator. Nanti mau minta autograph ini.
Fun guy. Yeah, I'm the fun guy.
Fun guy, yes. So thank you again to Dr. Ruben, and of course, thank you to our amazing line of speakers, Mr. Aminot Faiz Suis from FRC, my partner in crime, Miss Viviani, the fungi lady, and then Dr. Goh or Dr. Hubert Pitol on the birds, and thank you, Madam Louisa. Of course, thank you to Dr. Kartini from okay, and thank you to Boss John Sugao atau dalam ekspedisi we call him John Sugao, and of course to the lovely Miss Esther D. Kame. Thank you so much to all of the amazing line of speakers. So if any of you have any extra questions for all of the speakers and even for Dr. Ruben, our ecologist here, you can just email them anytime. So you can post your emails in the chat section to all the speakers. Who knows, some of the attendees or participants may have additional questions for all of your studies. So there you go, the morning session. For today's webinar, the fourth webinar focusing on the Mensalong Forest Reserve HoB Borneo Scientific Expedition. And we will have the evening session at 2 p.m. with another, I think, around 5 to 6 speakers also going to present on very interesting topics as well. So please, to all of you, stay tuned and we will see you again at 2 p.m. And before that, there's just some announcement. Is that okay now? Announcement. We will have a survey form, but it will be distributed after the end of today's webinar, which will be tomorrow. So please check your email, your respective email, the one that you registered with, and look out for the survey form. So kindly fill in the survey form and send it back to our email htan@sabah.gov.my. So we really appreciate if you can give us feedback on the webinar. So that is it for this morning session. Thank you, and we will see you again in the evening, which will start at 1:50 p.m. But I kindly request that all of you to please come if you can earlier than that, so that we can start as early as possible and to avoid any other disturbance, yeah, with internet connection and whatsoever, especially to the speakers for this afternoon, this evening session. So Dr. Arthur Chung will be the facilitator for this evening. We look forward to this evening session. And Bian, if you have any other additional housekeeping announcements that we would like to announce to everyone.
Okay, I just want to inform all the attendees for today. Please sit still and watch us this afternoon because there are more interesting topics to discuss. So please be ready at around 1:50 p.m. for a housekeeping announcement because we will begin our presentations around 2 p.m. So enjoy your lunch and thank you for your total focus on this presentation for this morning. So really appreciate it. So see you guys later.